Welcome everyone for our special event today brought to you by the Rappaport Foundation about aligning neuroscience and psychiatry for advances in our field of mental health. It's a great pleasure. My name is Kerry Ressler. I'm the Chief Scientific Officer at McLean Hospital, and I'm joined today by Professor Bill Carlezon, Christine Javaris, Galen Missick, Jakob Hartman, and Sabina Beretta. And I think we have a great hour of discussion planned for you, really talking about where the cutting edge of the field lies. But first, I wanted to just thank and appreciate the Rappaports and their foundation. Phyllis and Jerry Rappaport were founding members of the McLean National Council. They have funded research fellowships at McLean since 1999, starting with 1 and now 3 fellowships a year. And they're among our most steadfast supporters of research, understanding the potential within McLean's robust research enterprise and allowing their support to span many different diagnoses and modalities. Notably, this is over 23 years of support, at which time there's been transformational changes in neuroscience and discoveries in our field, from genetics to laser-guided optical control of cell processing in the brain in mice, extreme advances in genetics and large-scale sequencing and multiomics and machine learning, and the meaningful integration of translational neuroscience with understanding of human neuropsychiatric disorders is now really making great progress towards interventions. And that's what we'll be focusing on in our discussion with our scholars today. The Rappaport's foundation mission is to fund emerging leaders that's synergistic with McLean's mission to train the next generation of scientists and clinician researchers. The Rappaport grants have led to many McLean researchers receiving NIH grants, large grants, and other kinds of support to allow them to have sustainable funding and take this work really to the next level. Also, multiple generations of the Rappaport family are involved in the McLean family and McLean support. Their son Jim is now in the National Council as well. In December 2020, the Rappaport family chose to celebrate their longstanding partnership between the family and McLean by endowing McLean's basic neuroscience division and establishing a chair for its chief. Now the Phyllis and Jerome Lyle Rappaport Center of Excellence in Basic Neuroscience Research. And with that, I'll then hand it over to Professor Bill Carlezon, who's now the chief of the Phyllis and Jerome Lyle Rappaport Center of Excellence and the funded chair in psychiatry by this foundation, by this named Phyllis and Jerome Lyle Rappaport chair. He's the director of the Behavioral Genetics Laboratory and professor of psychiatry at Harvard Medical School. And furthermore, he's really just an international leader in translational research, leading one of the leading journals of the field, and really a leading thought leader in how do we go to the next step of understanding and translating our discoveries in basic model systems to human diseases. So without further ado, thanks so much, Dr. Carlezon.
Thank you, Kerry. Thank you for the kind introduction and the kind words. And I'm really grateful to be here today. So I'm Bill Carlezon, as Kerry just mentioned. And I'd like to start by just expressing my most sincere gratitude to the Rappaports for their generosity, not only to McLean, but to the broader field. I think everybody who's familiar with the Rappaports knows just how much they do, not only for psychiatry, but for other endeavors as well. It's just absolutely fantastic that they're able to sponsor events like today's event that get massive communities together and talking with one another. And while it's been a long time because of the pandemic, I'm looking forward to the day when I can see them in person again. So thanks again for all of your support. I'd also like to thank everyone today who is joining us through the magic of Zoom. Today, we're going to highlight an ongoing issue in the field, and that issue is that there are major gaps in the alignment of psychiatry and neuroscience, specifically with respect to work being conducted in humans versus model systems, such as rats and mice. The past couple decades have brought major technological advances in psychiatry, such as brain imaging, and in neuroscience, including some of the things that Kerry already mentioned, such as optogenetics and chemogenetics, which can be used to dissect with great precision the brain circuitry of motivation and emotional states. Still, there are a few examples where these improvements and advances have resulted in new treatments that are relieving suffering in people with psychiatric illness. As an example, there are very few instances of drugs currently used to treat psychiatric illness that were not repurposed as a result of serendipitous observation. So they were being used to treat something else, and they were repurposed to treat psychiatric illness. Even the latest breakthrough drug, ketamine, has been around for decades and has been used for other things, including surgical anesthetic for children and in veterinary settings as an anesthetic. So if there's a single take-home message today, it would be this: in the field of psychiatry, it would be a really good thing if we could make more interactions between people working with humans and people working with model systems such as rats and mice. So that's the overall guiding principle of today's discussion. Of course, we hope these things can happen organically, but we can also take steps to ensure that it happens. For example, by building it into our training programs as early as possible, as well as by including it in our grant applications. And I'll talk a little bit about that toward the end of my discussion. I understand that this premise may seem really obvious. Psychiatrists, clinical psychologists, and neuroscientists should be interacting, especially at a place like McLean, and it might be hard for people outside the field to believe that it isn't already happening. So why isn't it happening? I think the reality is that psychiatry, clinical psychology, and neuroscience exist on parallel tracks. Everyone is so busy and so focused on their own work, it feels hard to imagine that we have to take on even more than we're already doing. As an example, one of our panelists, Dr. Javaris, has to leave right at 5 p.m. Because she has a research subject scheduled for testing. In my own case, as an experimental psychologist who works with animal models, I've managed to get pretty far down the line in my career trying to model neuropsychiatric conditions such as substance abuse disorders, depression, bipolar disorder, and autism before I actually had the opportunity to learn more about how these conditions actually look in people. Everything I knew came from reading books or papers that were in the literature. Being based at McLean has changed all that for me, but few people in my field have had such a wonderful opportunity. I think that the systemic lack of exactly these types of experiences has led to many disappointments we've encountered when trying to translate discoveries we've made in the lab, in basic research labs and animal models, into new treatments that will actually help patients. So as one example, a psychiatrist can share firsthand observations that would be really helpful to neuroscientists. In our own line of research, one example is a common feature of autism is autoimmune dysregulation and seizures, especially during sleep, even though that these are not diagnostic features that someone like me would find in a textbook. Likewise, neuroscientists can be enriching clinical insights, such as while drug X looks like it might be treating the signs of condition Y in people, it looks like it's aversive to animals or making them sick, meaning that humans and patients might not like taking them, leading to adherence problems. Or that we should be more carefully looking at the brain activity, the activity of brain region X as a potential biomarker of condition Y. So one solution is to provide resources for training and activities across a spectrum, supporting psychiatry, psychology, and neuroscience, and building in new opportunities for collaboration. So this is one thing that the Rappaport family philanthropy does so beautifully and does so effectively. Today, we'll be hearing from McLean researchers who have been past Rappaport scholars and now represent established and emerging leaders in their respective fields. So without further ado, I'd like to ask each of our panelists to introduce themselves, to talk briefly about when they were a Rappaport scholar, and as an icebreaker to stimulate discussion, give us an example of an amazing brain fact. And to the audience, if there are questions, please add them either to the chat function or raise your hand, and we'll do the best we can to get to them. I also want to mention that we've set some time aside at the very end for questions and discussion. So I'd like to start with Sabina Beretta. Could you introduce yourself?
Sabina Beretta, I am an associate professor of psychiatry and in neuroscience at Harvard Medical School. I direct a laboratory here at McLean Hospital, the translational neuroscience laboratory, and I'm also the director of the Harvard Brain Tissue Resource Center. The first thing I'd like to say very briefly, thank you everyone for taking the time to attend this webinar, but particularly gratitude goes, of course, to the Rappaports that have made all this possible. I was lucky enough to get a grant from the Rappaport Foundation a little more than 20 years ago. I'm afraid that dates me a bit badly, but that's what it is. I would love to discuss more our work. My group works mostly on human postmortem samples, so we have our set of considerations on how to link what our findings, our hypotheses are on one end to animal models, on the other end to clinical research. But for the time being, a fun fact about our work, a recent one in collaboration with several people actually on this particular webinar, is that we can actually culture live cells from postmortem human tissue, specifically different types of glia, and I'll be happy to discuss that later on.
That is the most, still for me, the most fascinating thing I've learned in a long time. So thank you, Sabina, and welcome. Next, Dr. Kristen Javaris, Assistant Professor of Psychology in the Department of Psychiatry at Harvard Medical School.
I'm Kristen Javaris, and I'm a clinical psychologist and statistician by training. And both my research and my clinical practice focus on eating disorders. And in particular, in the past few years, I've started to focus on understanding more about how stressors and negative emotion impact eating behavior, especially in eating disorders. And I'm very, very grateful to the Rappaport Foundation because they played a very crucial role in helping me start that work. And I'll be telling you a little bit more about that today. But for now, my fun fact about the brain is that your brain responds differently to food if you're hungry compared to when you've just eaten.
Thank you, Kristen, and welcome. Next, Dr. Jakob Hartmann, instructor in psychiatry at Harvard Medical School.
Hi, I'm Jakob. Yeah, I am an assistant neuroscientist in Kerry Ressler's lab at McLean, and an instructor in psychiatry at Harvard Med. I would also like to take this opportunity to thank the Rappaport family and foundation for the fellowship that I was actually being awarded this year and for the opportunity to speak here today. As you may hear, I'm originally from Germany, where I studied neuroscience, and I did my PhD at the Max Planck Institute of Psychiatry in Munich. And I joined McLean Hospital in the fall of 2015. An amazing brain fact that I thought was very interesting when I first heard about it when I was still in college was that in neuroscience, you can actually stimulate brain cells of mice with light to drive their behavior. And by applying this technique, which is called optogenetics, you can, for instance, increase or reduce social behavior or anxiety-related behavior in mice. And yeah, very broadly speaking, my research focuses on to better understand basically the behavioral and molecular basis of individual stress vulnerability and resilience in the context of psychiatric disorders.
Thank you, Jakob. And last but not least, Dr. Galen Missig, instructor of psychiatry at Harvard Medical School.
Thank you. So I'm Galen. I'm an assistant neuroscientist. I work in the behavioral genetics laboratory run by Bill Carlezon. And so before I begin, thank you for the invitation and thank you for the support. I was a Rappaport fellow in 2017 to 2018. And I kind of studied the interactions between the immune and the nervous system and what this all might mean for psychiatric conditions. And for my fun brain fact, it's not really a brain fact, but I spend a lot of time actually trying to make the data you can get from a smartwatch the same kind of data to measure that in a mouse and a rat. So it's more of a fact, an effort than it is a fact, but that's what I'm going with.
Thanks, Galen. It's not a brain fact, but it sure is fun. I can attest to that. So thank you. And welcome to all of you. So what I wanted to do next was to go through some questions and encourage each of the panelists to respond. And they're all geared to think about how the training has influenced your career and your career trajectory, but also woven in there, if you could think about, you know, the today's premise is how can we improve things? How can we improve communication between psychiatry, clinical psychology, and neuroscience, and the consequences of that, you know, the benefits for the field that would be the result of that. So I guess the first question, and some of you have already answered it a little bit, but can you tell us a little bit about the work that you did that was funded by the Rappaport Foundation? Does anyone want to volunteer to go first? Sabina, maybe?
Yeah, I'll go first. I was actually just last night went back to the slide deck that I had presented as my progress report to the Rappaport Foundation, and it was really striking to me that, in fact, those findings that were a bit more than 20 years ago really started a field in my laboratory that has continued until now, has current and past grants funded, eventually started a field that was picked up by many other research laboratories. In very kind of broad terms, we discovered that what we could call a sort of a third component of the brain that is very rarely, or at least until that time, it was rarely studied, which is the sort of set of molecules that surround all cells in our brain and guide the way we learn, the way we remember things, the way our brain develops. And we discovered, as a consequence of that particular fellowship, that they are disrupted in the brains of people with schizophrenia and bipolar disorder. And that led us to a long, long path that we're still pursuing on trying to understand what are the causes and consequences of these changes we found in this particular set of molecules. The challenge is to understand what are the, on one side, the symptoms of that are sort of supported by these changes. In other words, how do those changes eventually show in the behavior, in the symptoms of people that have those disorders, schizophrenia or bipolar? And on the other side, how can we mimic them in models that are relevant to these disorders so that we can understand their causes? This has been particularly challenging. We are doing it in many different ways. One has been looking at animal models in which we try to cause the same changes and try to understand what are the consequences, for instance, for their brain development and from their plasticity. We also have had interesting ways to look at them. For instance, another fun fact that we could share is that there is one source of neurons in us that is actually accessible for biopsy. And these are neurons that are in the ceiling of our nose and are those that allow us to smell. And there are a set of neurons and glial cells that eventually connect to our brains, but they can be biopsied. So another way to do it is, in fact, to collect those from biopsies and from people that do suffer from these disorders, and try to understand how in those samples these changes are affecting the functions of these cells. I'll leave it at that for now, but again, it was really, truly both striking and touching to me to see how far we have gone with that, with those findings that were funded by this fellowship such a long time ago.
Thank you. It's fabulous, and thank you for sharing that. Kristen, would you like to go next?
Sure. So I'm going to give you all a little bit of background first. And so one thing that we have learned is that interpersonal stressors can precipitate fairly marked changes in eating behavior for some people. And so for some people, they might have a negative interpersonal experience, and it might make them want to eat more, potentially even engage in binge eating. But for someone else, they might have very similar negative interpersonal interaction, and they would actually want to eat less and potentially even restrict their dietary intake. And so I was interested in the mechanisms that underlie these divergent responses in eating behavior. But I first sort of had to take a step back, because before you can test any hypotheses about mechanisms about a phenomenon, you have to be able to measure the phenomenon. And so one thing that I knew was that people's own report of their eating behavior doesn't tend to map on especially highly, or correlate especially highly with more objective measures of their eating behavior. And so I knew I didn't want to rely on self-report, because I was more interested in actual objective changes in eating behavior. So the Rappaport Award really gave me the opportunity to get into the laboratory and to spend time developing and doing some preliminary testing of a task that assesses how a particular type of interpersonal stressor impacts people's intake of chocolate milkshake, which tends to be the type of food that people crave when they've had negative experiences. And so one thing that we found in doing this was that people's report of how they would react under stress, or how their eating behavior would react under stress, had effectively no correlation with how they actually reacted during the task. And that actually is something that's replicated in a large number of data sets over the past few years. And so that sort of adds to the evidence that people's perception of their own eating behavior can be very different from more objective measures. And that's a lesson that I've taken forward with me in the research I've been doing since then.
Thank you. And, you know, maybe we can discuss this later, but that's really important, that you need this subjectiveness in addition to the objectiveness that many of us are striving for right now. So the same people see the same thing. Two people looking at the same phenomenon in the lab need to be able to see the same thing. And we consider that objectivity to be really powerful. But I think what you're telling us is that we can't lose sight of subjectiveness either. So that's important. Thank you. Galen, would you like to go next?
Sure. So try to give a brief context. So for a long time, we've had an inkling that there is a potential connection between the immune system and psychiatric disorders. We've seen things like elevated immune signaling, both in the periphery and the brain. And there's been some genetic studies that have kind of identified a number of immune pathways. But what's really come to light in recent years is just how interconnected the immune and the nervous system are. So for example, some of the same molecules are actually being used by those systems. And we're finding more of these connections kind of every day. So for instance, the same molecule that eliminates invading microbes is also going to be used to eliminate connections, or those synapses in the brain. Anyway, the reason why that is important is because now you can imagine there's this situation where, you know, during some key development of the brain, so you can imagine the embryonic brain while it's developing, if you give something like an infection or something that stimulates the immune system, you may be able to alter the trajectory of the developing brain and result in something like a psychiatric condition. And you know, we've known that there is some epidemiology for this. So for instance, if you look at offspring of women who have had various forms of infection during pregnancy, those offspring are at increased risk for a number of psychiatric conditions, including depression, autism, and schizophrenia. Anyway, so what I do is I attempt to model this in the laboratory in a highly controlled way. And what we do is we use a model system, in this case, mice and rats, and we're able to deliver an immune stimulus at key periods and look at how it affects that developing brain. Now, I was not the first to do this. There have been many before me. And what they discovered is that if you give an immune stimulus, you can produce this kind of phenotype where they're kind of like a little bit less, the animals, of course, and this is an important point of discussion. The animals are a little bit less social, have heightened anxiety-like behavior, and exhibit a number of other changes. And so one part of my research is to really look at this model and to use some kind of new measures, interest in things that we term a little bit more translational, or things that are a little bit more similar between animals and humans. But what the Rappaport Award allowed me to do is to investigate this kind of new idea that kind of I had, and it was really looking at like, all right, what is the assumption that is being made in this model? And it was that this immune stimulus that we were using was really perfectly capturing this environmental risk. And we knew that there is an incredibly diverse array of immune pathways. And so I tried out another type of immune stimulus, thinking I might get, a more effective model. And sure enough, I got something completely else. I found that it was actually a very different phenotype. And in fact, in some ways it had actually more social behavior, but it was kind of disrupted social behavior, opposite changes in anxiety, really just a plethora of different changes. And the main takeaway was really that, so not only can we modulate the nervous system by giving these immune stimulus, that depending on the different types of immune stimuli, you can alter the nervous system in different ways. And this may provide really kind of some evidence for why we have this one global kind of risk factor of the immune activation being associated with kind of this wide array of psychiatric conditions.
Great. Thank you so much.
Sorry for the mile a minute, but
thank you. Jakob, I know that you just started your fellowship, but if you could just tell us about the general premise and what you'll be looking at.
Yes, of course. So when you think about mood and anxiety disorders, it's well established that they are that they develop as a result of the interaction between genetic predispositions and environmental risk factors such as trauma or chronic stress. But as with other complex illnesses, there is no universal susceptibility gene for such disorders. There's no gene for depression or for post-traumatic stress disorder and so on. Instead, it is believed that multiple genes with small effect sizes may actually contribute to the disease phenotypes. Moreover, there's only a subset of individuals that is actually susceptible to stress or trauma, while many others can show a remarkable resilience to adverse experiences. Now, with my Rappaport Award, I'm trying to better understand the behavioral and molecular basis of this individual stress vulnerability and resilience in the context of psychiatric disorders. In my research primarily focuses actually on mice. The advantage of working with mice is that we have access to their brains at the end of the experiment. So even when you chronically stress mice, they can develop a similar phenotype, for instance, to humans. So they can also show an increased susceptibility to stress, which would be reflected in, for instance, an increased anxiety-related behavior or increased social avoidance or cognitive deficits. Could also be that they show an increase in circulating levels of stress hormones, for instance. But you would also observe a subset of mice that shows some sort of a resilient phenotype. So they basically behave pretty much like unstressed control mice. And now what we're trying to do is we want to investigate the brains of resilient mice, of stress susceptible mice and control mice, And we want to investigate their molecular profiles of a specific brain region, the bad nucleus of the stria terminalis, BNST. And with a fairly novel technique, single cell RNA sequencing, we're trying to identify resilient specific molecular signatures that we basically only see in the resilient brains, but not in the susceptible brains or control brains. And so there we're collaborating with another lab at McLean from Nikos Daskalaskis, who's helping us a lot with the bioinformatics analysis. And based on like the transcriptional profiles that we would see in these resilient specific subpopulations, we could then basically try to, if we find, for instance, that there's a certain cell type that is extremely active in the resilient animals when we stress them compared to susceptible animals, then we could try to introduce this molecular signature into a new batch of animals and see whether we can shift the behavioral outcome from more susceptible animals to more resilient animals. And this we would do, for instance, with techniques like optogenetics or chemogenetics, so that we can really either increase the activity of neurons and their function, or we can completely silence them.
Thank you. It's really interesting work. And one thing to emphasize, you said it, but I'll reemphasize it, is just how much data, how big the data sets are becoming. No ordinary human can analyze these data sets anymore. You need incredible computer power that can make associations between all of these different individual genes and sets of genes. So it's becoming really, really complex, and we need additional tools to solve these problems. We call it, it's called big data in the field. That's what we call it as a shorthand, but it's becoming increasingly important with all of the experiments we're doing. So thank you. Keeping in mind that one of the premises of today's meeting is to think about better aligning psychiatry and neuroscience, I'd like to ask each of the panelists if they could give an example of how their own line of work might benefit from better alignment of psychiatry and neuroscience. So Kristen, do you want to start with this?
Sure. So I'm going to give an example that doesn't pertain to my particular area of research, but is relevant to eating disorders more generally. And I wanted to bring this up because it's an example of something where, at least for me, it's a little bit hard to think about how you would align psychiatry and neuroscience. And maybe with all of the neuroscientists here, you'll have some insights. And so in eating disorders, there are behavioral symptoms, so like restricting your dietary intake, binge eating, and there are also cognitive symptoms. So one example would be putting a very high value on being at a low or lower body weight, and being at a low weight have a very heavy influence on your evaluation of yourself. And so those cognitive symptoms, I often term them sort of sticky. They can occur before other eating disorder symptoms emerge. And oftentimes they linger on after the eating disorder behaviors are remitted. And they can be very, very distressing. And I've had many clients I've worked with say to me, if I could just get rid of the thoughts, it would be so much easier to change my behaviors. And they often do persist for a long time after the behaviors have changed. And so, for me, at least, it's a little bit more straightforward to think about how, in at least preclinical models, you would model some of those eating disorder behaviors. I myself haven't really come up with a way that you could necessarily model some of the cognitive symptoms of eating disorders in animal models, because we hope that rodents are not feeling negatively about their body image or putting undue influence on their weight or shape. There are some ways that I've thought about, for instance, disruptions in people's perception of their size that could potentially be modeled in animal models. But I give this just as an example of an area where, at least for myself, I haven't quite figured out how to align psychiatry or psychology with neuroscience.
Well, what you just said, I think, was very informative for me. You mentioned attributes that seem related to impulsivity and compulsions, and we know from animal models what parts of the brain controls those types of thoughts, cognitive function. So even the brief description you just gave has given me, as a neuroscientist, a whole bunch of different thoughts about how we might take small attack the problem from different perspectives. So that was fabulous. Thank you. Sabina?
So I'll give an example of related to our work, although it's a bit in general. So what we try to do is try to understand what kind of brain changes from the point of view of molecules and cells are in the brain of people that suffer from psychiatric disorders. The problem we have is that the psychiatric disorders have a very complex clinical presentation. Even disorders that we consider in general terms as categories, such as schizophrenia, bipolar, are very complex and very heterogeneous, have a multitude of different presentations. You could say that no patient is really like another one. So when we look at many different brains, and we see many different changes at the level of, again, different brain regions, different parts of our brain, their components, how do we link them to the complex set of symptoms that these donors had? And that has been something that I believe has been a question for as long as the field has been evolving, that we are trying to solve in many different ways. Again, you really kind of doing work on postmortem human brain, you kind of try to bridge between animal models and clinical on the two sides, but both are difficult, although they are making strides. So an integration, for instance, and we are trying to do it more and more. For instance, McLean is a great place to do this because basic neuroscientists and clinicians, and oftentimes clinician researchers, are all in the same place, all interested in the same topics and in asking the same questions. But we try to work more and more and with clinicians asking, how do we go from a specific molecule in a specific brain region and think about what was the underlying symptoms, or what was the resulting symptom rather. And this has been something that we think a lot about. We don't have perfect answers, but it is an evolving field and a very interesting one to us.
Thank you. Jakob?
Yes, I fully agree. I think a better alignment of psychiatry and neuroscience is extremely important. I think this was also one of the reasons back then that I chose to join Kerry Ressler's lab, because he really focuses on translational research, bridging molecular neurobiology and animal models with human genetic research. And what I like so much about being in his lab is that there's a lot of crosstalk going on between the folks in the wet lab that are working with mice or with cell cultures, but also with the people from the human genetic side. And we have even a few MDs and practicing psychiatrists that also work as researchers in our lab. And I think that makes his lab, but also the entire McLean Hospital, such a special place to work at. And not a lot of research settings like this exist actually around the globe. That being said, we definitely need better animal models to better understand psychiatric disorders. I think there's been a trend in the past few years for implementing, for instance, supervised machine learning approaches into the behavioral analysis of our models. that really enable us to detect even very subtle behavioral differences that we would usually not even recognize with our bare eyes, or just because there's it's just so labor intensive that we would not succeed to score all those videos. Basically, now with supervised machine learning, that goes a lot, actually a lot faster. And I think this is just the beginning. So in the future, such approaches actually may help us if we, for instance, just monitor the behavior of mice in their home cage, their activity, that we would be able at some point to predict which animal will turn out to be resilient or susceptible before we even subject those animals to stress. And that might shift the field again in or fast forward our research. But definitely it's challenging. We cannot ask a mouse how it's doing, how it's feeling. We cannot model suicidality or something like that in animal models. And yeah, it will stay a challenge.
Thank you. And just so we're all on the same page, machine learning is a phenomenon that can, just to boil it down in this particular instance, it can detect not only individual behaviors, but patterns of behavior. So it can do analyses on, before the animal did this, it did these 10 things in a separate order, and it always does these 10 things. It's a method of getting at these big questions that an individual person would never be able to detect. You really need this computing power to see the patterns. So thank you, Jakob. Galen?
Yeah, I mean, I completely agree with the other panelists and Jakob. I think this is one of the major challenges of our field. Is really, I mean, as Kerry said, like, he made this big list of all these amazing things we've developed. It was even since I was an undergrad. Like, we are now master manipulators of the nervous system, turning off, on, we can measure hundreds of neurons at a time. But what really hasn't gotten the same kind of boost is the endpoints, right? Take your model system, your mouse and rat, you can do all these manipulations, but then what do you read out? And that hasn't changed nearly as much as all these other processes. Oftentimes we're doing things like you put a mouse on a little maze, and some of the mazes are open and some are closed, and you see how much the mouse spends in the open versus the closed arm. But you're not going to do that with a person. You can't do these same exact tests, and who knows what that mouse is thinking. It comes down to, we were always just kind of anthropomorphizing this. I think in this particular situation, the mouse is probably scared. I would love to have better measures in this. It's just, we don't currently, unfortunately, and I think it's a very tough area. And it's easy to, go down the rabbit hole. We have all these great techniques, so let's just keep going with the good ones we have. But I think if, making that leap from a model to people is still a huge barrier.
Great, thank you. And I agree 100%. And you mentioned at the beginning, what we're trying to do here is to align the endpoints. So you pointed to your smartwatch, and we can start measuring those same types of endpoints, like things like body temperature and activity and patterns of activity, even sleep. So we're comparing apples to apples and oranges to oranges for maybe the first time in the history of psychiatric neuroscience. So thank you for your answer. So we've got about 15 minutes left. I wanted to save time at the end to answer some questions, to have free Q&A. I do see that there's a few questions in the chat, and my intention is to get to those first. But I'd like to try to wrap up here with a couple more questions, and hopefully you could answer them more briefly in this case, so we can get to the Q&A. But my next question would be that once psychiatry and neuroscience are better aligned, what other fields do we need to start thinking about drawing into alignment with us as well. So Jakob, do you want to start with that?
Maybe we should look more into the relationship of the gut microbiome and the brain function. Actually, I think this is a topic that is also getting increasingly more attention. There have been repeatedly studies now showing that there's a bidirectional communication between the brain and the gut that involves neural, hormonal, and immunological pathways. And in the end, maybe this microbiome gut-brain axis might also provide novel targets for the prevention and treatment of neuropsychiatric disorders in the end.
That's great. Great point. Galen, you want to go?
Sure. So, I think one of the things we need to look to and try to look to more is kind of ecology and kind of animal behavior and really understanding the conditions in which, the brain may have evolved. I'm a firm believer in my own ignorance. I don't think I can truly understand, the conditions of, these primitive humans in which the brain has actually, been adapted to. It's so hard for me to understand that I think there's probably some kind of element there that we don't fully appreciate.
Interesting. Kristen?
So I may be slightly biased, but I think we could benefit from embracing some of the approaches that epidemiologists use. And so in particular, epidemiologists tend to have very large samples, and they tend to have representative samples. And that's not always true in some of the work that we do in psychiatry and neuroscience. And I think it would be really interesting to start looking at some of the mechanisms that we've identified as being associated with disorder in these larger population samples, because you may find that some people have those mechanisms but don't have the disorder. And that may give you some insight into ways to prevent the disorder or treat the disorder. And so I think there's a lot of power when you have these very large samples and when they're representative of the general population.
Thank you. Sabina?
One obvious one that we referred in passing, but I want to make sure that it is kind of more centerpiece is obviously genetics. We know that all psychiatric disorders have a very strong genetic components, and yet one of the holy grail and the most difficult thing to do is to go from genetic vulnerabilities through what happens to the brain as a consequence and on the way to symptoms. So it's an obvious one, but I just want to kind of make sure it's on the table. The other one that is probably kind of a more generalized way to say things that have been said already is, for the longest time, it seems that our field focused on the brain as if it were separate from everything else in the body, that it wouldn't be affected by anything else that was happening. We know it's not true, and more and more we have evidence that there is a lot more communication than we thought. Immune system, microbiome, and many other things are very obvious examples, but there may be even more that we are not even realizing. And there is a lot more communication between the brain, which again, at the beginning or until not very long ago, let's say, was seen as a sort of ivory tower, kind of impervious to anything else that was happening to the body. And now we know it's not true. So in fact, the whole general medicine and human body physiology would probably at some point be integrated into psychiatry.
Thank you. Those are great points. I'll answer this one too. I would like to advocate for engineering. I think a lot of the newest techniques that we're using are derived from engineering. I personally would love to get EEG recordings from a smartwatch or a device like that, and that may be possible in the future. I also think the way that engineers think about things is that any problem can be solved, and biology just becomes a different type of engineering problem. So, you know, I think the field is already doing this and can benefit from more embracing of engineering. So I want to ask one final question before we move to Q&A, and if the panelists could just answer this with only a few words or a few descriptions. But as emerging or already established leaders in the field, what can you offer as a couple of words of advice to the next generation on the secret ingredients of how to be a successful scientist? So Galen, I'll start with you because I don't think you've gone first yet.
Okay. Oh gosh, I was going to wait for the other panelists, honestly, and I wish I had those secrets. Honestly, I think things that kind of helped me is just always keep in mind the bigger picture. You know, see where your research and things align with, you know, the big picture of what's going on. It's very easy to get kind of bogged down into the details of a particular experiment or your line of research, but it's always important to remember.
That's great advice. Sabina?
Well, one very brief is a recommendation that one of my mentors when I was a postdoc gave me, which has helped me every time I found something else that was interesting and I wanted to follow it. And it was ask a question and follow it and keep following it. Don't get distracted. And of course, our thinking evolves, our methodologies evolve, but science is so interesting and there are so many things we want to do that reminding myself of that advice. Ask a question, make sure it's an important question, stay with it. But the other one, very, very briefly, is our field has evolved and researchers can no longer work as an island. The only good research that it can be done at this point is research that is very highly collaborative and possibly between fields, which is in many ways what we are discussing. Pretty much everybody on this panel collaborates with pretty much everybody else and many, many more. And that is really important, not isolating oneself, trying to build a team, try to expand it, try to include competences that you don't have is probably one of the most important points.
Thank you. Kristen?
So I guess one thing I would say is it's important to have perseverance because I always joke that in science you're stuck most of the time because when you're not stuck, you move very quickly until you get stuck again. And so you have to, be able to persevere through obstacles. And the other thing is you really have to, it's helpful if you can view constructive criticism as a chance to make your research stronger and better. Because it's inevitable that as a scientist and as a researcher that you're going to get criticism of your work, and I think that the most helpful way to approach it is really by thinking of it as a way of making your research stronger and helping you think about things even more clearly.
Thank you. And Jakob?
I agree with all the points the panelists have already said. I would add, for instance, especially when you're still an early career investigator, it's important to really find a good and supportive mentor. If you work in a fancy lab, but you don't have the support from your mentor, then the next career steps will be, or potentially might be more challenging actually. So always keep that in mind when you choose your next lab, that you have a great mentor. And maybe another thing is that you should try to make your research publicly available, either by publishing open access, which of course can sometimes be more expensive, but you could also try to upload your results or your manuscripts to a preprint server. Like that, a lot more people will actually have access to your studies. A lot more people will read your papers and will cite your papers, and that in the long run will also help to get more funding, actually, to do more research.
Those are great points. Thank you. And thank you to all the panelists for answering those questions and giving them careful thought. In the last couple minutes, I'd like to try to answer some of the questions. And Kristen, I know that you have to leave at exactly 5. So there is a question that I'm going to take out of order. It's not the first one that came in, but it's for you. I'll read the question. Do you think there's room for translating the more behavioral aspects of eating disorders, such as food craving or avoidance, almost like addiction or compulsive behaviors?
Yes, I definitely do. So I didn't mention this, but there are existing animal models, for example, for anorexia nervosa. So there's the activity-based anorexia model in rodents, and it nicely captures some of the same features that you see in anorexia nervosa. So for instance, adolescent female rodents are more susceptible to the activity-based anorexia paradigm. They're more likely to stop eating in that paradigm, which is, we also know that adolescent females are at especially high risk of developing anorexia. So there are models, and you can, there are definitely people who have developed models for the various components of addiction and applied to food addiction. In fact, there was a great review article on that in Neuropsychopharmacology a few years ago. So I definitely think that people have already done a lot of great work, and there's room for a lot more as well, modeling some of the more behavioral aspects.
Thank you. And I know that you have to drop off. So in case somebody's in the middle of an answer, I learned a lot from listening to you today, from meeting with you. And thank you so much for your time and your insightful answers.
Oh, my pleasure.
Next question is, why hasn't schizophrenia been classified as a neurodevelopmental disorder in the DSM? Do we agree that it should be? Or does anyone have thoughts on that? Sabina, maybe? Or Kerry?
I definitely agree. It should be, schizophrenia should be treated, and in fact, from a scientific point of view, is treated as a neurodevelopmental disorder. I would argue that the same can be said for several other psychiatric disorders that have, may have an onset in adolescence or even early adulthood, but in fact, we now have prodromal syndromes and changes that occur much earlier on. I'm not one much for science politics, so I'm not going to go into the question of why it hasn't been included into DSM as a neurodevelopmental disorder. The only comment quickly that I would make is that the obvious one, autism is often diagnosed much earlier, at earlier age, and therefore that's why it gets treatment earlier. It is very difficult to make predictions, even on kids at risk or high risk, on whether they eventually will become full, will develop a full syndrome of schizophrenia or other disorders. So that complicates things from a sort of prediction point of view and knowing when they could be treated. But I completely agree with the developmental part of it.
And I would just add, I would agree with everything Sabina said. I think part of the issue is DSM-5, unfortunately, in our entire history of nosology and psychiatry hasn't been scientifically based. It's been epidemiology and consensus based. We do hope that in the next iteration, as work like that's going on here goes on, we'll be able to have more neuroscience based understanding of these disorders. And I think, as the second point Sabina made, is there's not really a developmental home for many of our adolescent onset disorders, and you could argue many of them are depression, anxiety, substance abuse, etc. So I think there's great room for progress there. And also, I think as a field, we have a lot to learn from the autism community. I think our allies in mental health, we've not been able quite to break through in the same way that autism speaks in others. And hopefully with decreasing stigma and other things like that, we'll be able to make more public awareness for the importance of research in all these areas. So thank you, Sabina.
It's 5 o'clock. I want to try to slip one more question under the wire, just because I think it may be a 1 or 2 word answer. And the question is, how similar are immune systems in rodents and humans? Are they similar enough to draw inference for humans? My answer would be yes.
Mine would be the disappointing yes and no, where, in some cases, yes, they have shown a lot of similarity, and in other cases, they're not at all similar.
Thank you. With that, I think I'd like to wrap up and thank everyone. The Rappaports, Sean for organizing this, Kerry for your wonderful introduction, and all of the panelists, Kristen, Galen, Sabina, and Jakob. Again, I learned a lot today from being part of this, and I really appreciate you taking the time to be here today. So, and the audience as well, thank you for joining us. Good afternoon, everyone.
Bye, everyone.
Thanks, everyone. Thanks, Rappaport Foundation.